in already for our webinar on critical toxicity information for early R&D decisions. And we have some excellent speakers today. So my name's Laura. I'm going to give you a brief welcome, and then we'll hear from Neelam Vaidya. So we're really pleased to have two wonderful speakers today, and you'll hear from each of them. And we will have time for Q&A at the end. And we are recording this talk, so people who cannot attend in person can listen later. So right now I'm going to turn it right over to Neelam, the CEO of Viridischem, for a brief hello.
Hello, everyone. Thank you for joining the webinar. This is as you know, this is part of the webinar series where we'll be discussing green chemistry, what it means for the different industries. This is the second webinar. We will be having at least 5 more webinars on the topic from the presence from the different industries. So before we go further, I would like to introduce our wonderful marketing team that is making these webinars happen and manage them very smoothly. Laurie Kretschmar, you have already seen her, and behind the scenes is Riddhi Dadda, who is making sure everything is working smoothly, and she'll be also posting some questions for you to answer from time to time, just to make the webinar a little bit more fun and interactive. So without further delay, Laurie, take it over.
Right, thank you. Thanks again, everyone, for attending. We're so glad to have you. And we have some wonderful speakers today. First, we're going to hear from Kirsten Ruslow. She's got her own consulting firm. It's called Process Profiles. And she's a chemical engineer who helps her clients really understand and improve their environmental impact. She provides them with all kinds of information to help them identify the products, processes, and substances that will be the most appropriate focus for improvement efforts. Then after Kirsten, we'll hear from Dr. Patrick Harman of BASF. He's an industry manager and responsible for regulatory affairs and advocacy, sustainability, and innovation. He has a strong interest in green chemistry and sustainability. And you'll also hear a little bit more from Viridischem's CEO, Neelam Vaidya. And first, now we're going to start with Kirsten Ruslow. Thank you, Kirsten.
All right. Thanks so much for Viridischem for inviting me to speak today. This will be quick. I only have 15 minutes, and then it's going to be Patrick Harman's turn. So normally I prefer to be interrupted if someone has a question, but this time we'll be doing questions and answers at the end. So if, or maybe I should say when, I don't do a good job of explaining something, we can come back to it, and you can type your questions into the chat as we go along so that you don't forget what they are. Before we move into my talk, though, I want to know how many of you have done process development where toxicity was considered at all stages of the development. There should be a poll button at the bottom of your screen now, and if you could click to take that poll, that'd be great. You'll get to see the results at the end of the presentation. Thanks for taking the poll. So the takeaways I want you to have from my talk is that there's very little opportunity to reduce toxicity after the development stage. The values you need to evaluate toxicity might not be readily available, but you can create procedures to compensate for that. And taking the time to consider toxicity in the early stages is prudent. Oops. I could have chosen this is my outline. I'm going to give an example of what can go wrong, and then talk about green chemistry and where to incorporate it and what the trade-offs are. And then we'll go on to how to evaluate the toxicity of substances. Talk about one way is using lists of problem chemicals. Another way would be using the globally harmonized system of classification and labeling. And I'll go through an example framework that, isn't the framework to use, but it's an example and an example comparison. I could have used another example, but PFAS will do. They're persistent. They're forever chemicals. They're very resistant to degradation in the environment. They have multiple carbon fluoride bonds that are very stable. They're bioaccumulative. Once they're in the body, they have a half-life that's on the order of years, and they're toxic. They're associated with a lot of health effects, including thyroid hormone disruption, increased obesity, cancer, and negative effects on the reproductive system. So one thing to know is that if something's both persistent and toxic, it really doesn't matter if it's present only in low concentrations. So this is a fairly loose and simplified storyline, but it captures the gist. Teflon was discovered in 1938 by mistake. They found out that it could be produced in bulk by using PFOA as a precursor. They developed a process for making PFOA, and I wasn't in the room, but I imagine they considered the traditional things: operating and maintenance expenses, yield, purity, and throughput, capital expense. And then in 1951, they started making PFOA on a commercial basis. And they realized that they were going to have to dispose of PFOA containing waste. And they decided to do that either by incineration, which is tricky because of that carbon fluoride bond, or by storing it in drums that, you know, are 100% likely to leak just given the right time frame. Then in the 50s and 60s, they found out that PFOA could be toxic to humans. Along the way, they discovered other uses of PFOA where it was an ingredient in products that got used so that it wasn't all getting made into Teflon. In 1998, they found out that PFAS are in the blood of just about every American. And then in the early 2000s, most PFOA was removed from consumer products. So it wasn't showing up as an ingredient in consumer products anymore. In 2009, they started making Gen X and then HFPO-DA as alternatives to PFOA. And just a couple of weeks ago, we learned that they have discovered that Gen X and HFPO-DA are perhaps more than 6 times more toxic than PFOA. So along the years, tens of thousands of tons of PFOA and other PFAS have been released to the environment in the US during product use alone. And releases have also been occurring from manufacturing facilities. So at the time that PFOAs were invented, you know, and other PFAS in the late 1940s and the early 50s, disposal and treatment were an afterthought. It was kind of an innocent time. But 2009 wasn't the dark ages of chemical industrialization. And this part happened during a time of heightened awareness. And it makes you wonder, what kind of blunders are we baking up now? Here's a chart that shows the stages at which green chemistry can be considered. Is this the right product if you consider toxicity? Is it the right pathway to that product if you take toxicity into account? Is it the right process for that pathway if you consider toxicity? Is this the right way to use the product if you take toxicity into account? And you've already been thinking about treatment and disposal because you know what you're going to be making. So a major barrier to including toxicity during process development is that toxicity values are uncertain. And it's true they are, but we've accepted uncertainties in things like future process performance and uncertainties in the market and in the cost of reagents over time and in the time value of money during process design. And if we can accept those uncertainties, we can accept the uncertainties in toxicity and other social impacts during process design too. This slide could be a 15-minute talk all by itself, but I'm just going to point out that the rule is you'll always have a trade-off. You'll always be faced with a trade-off. You're not going to be choosing between good and evil. And even if you just limit the universe of considerations to toxicity impacts or one type of toxicity impact, which you would never ever do, you're still going to have trade-offs. It's simple but insufficient to consider only lists of bad actors or lists of good chemicals, partly because When you do that, you're ignoring trade-offs. And also because the lists change, chemicals get removed and added. There's a list of okay substances called clean ingredients, and 38 substances have been removed from that list. So if you were banking on that list when you made your choices and they took your choice off the list, then what do? Lists don't allow you to be proactive and appropriately evaluate chemicals that just haven't happened to make their way onto a list yet. And the lists can be capricious. Some of them are based on quantitative evaluations of data that has been curated in a transparent way, so they aren't capricious, but others aren't transparent at all. You know, it's just a chemical on a list. A lot of the lists have a particular health focus, you know, some effect that they're tuned into, and they're blind to other health impacts. One of the main things, though, is that bad actors aren't necessarily on the lists of bad actors. So if something isn't on the list, doesn't mean it's good. And if you find a substance that's not on a list, you have to treat that as a data gap and not as data. But even though there are a lot of pitfalls with relying on lists, there is some information that you can glean from them. They're not totally useless. I'm going to briefly describe GHS here, because after this, I'm going to show you an example toxicity evaluation framework that relies on GHS assignments instead of on lists of chemicals. So REACH in the European Union and OSHA in the United States require manufacturers and importers to evaluate the hazards of the chemicals they produce or import. And they have to provide labels and safety data sheets so that downstream users have the information in a way that's aligned. It's the same for every substance. It's been created in a similar way. And a couple of the things that GHS includes are hazard categories, and their hazard categories start out with 1 as the most potent. So it's not like the safety diamonds where the larger number is a bad thing. And then hazard statements, so a lot of times you can see which bodily function is impacted. And then European harmonized GHS classifications are available for nearly 7,000 substances, which sounds like a lot, but it's really just a drop in the bucket. In my example framework, I start with European harmonized GHS assignments. And why would you want to do that? It's because these assignments are based on instructions that are numeric, and the choices of toxicity data are transparent for every substance. So if you were going to, like, here's an example framework. You decide on the impacts you want to include. So if exposure scenarios are limited, you can ignore transient effects. You want to think about how you're going to compare the different types of toxicity, acute versus chronic, reproductive versus carcinogenic, and on and on. And then develop a hierarchical framework for identifying hazards. So in my framework, I would start with the European Union harmonized GHS assignments. And if they're available, use them. And if they're not available, see if data is available and if they converge. And keep adding layers if you find a substance that seems favorable. So your first step might be to collect producer GHS assignments for substances that aren't EU harmonized. And there's a database of assignments that producers have provided to the European Union. That would be one place to go for that. You could look at safety data sheets. That's another place to go. And then and beyond that, you could convert raw toxicity values to GHS assignments because the instructions are available. And so to find those raw toxicity values, some of the lists on the list of lists have raw data with citations. You could look them up. You could just search literature for them. You could use proprietary data sets. And then there's a tool called TEST. It's the US EPA's toxicity estimation software tool, which you would never want to use for assigning benign status, but it might be a flag for a compound that you just could take off your list of considerations. And then if you have no data or not enough data, or if the data don't converge, you have to assume the worst. We're only going to look at toxicity here, not any of the other environmental safety or safety hazards. So let's assume we decided to ignore transient effects. So we're not going to look at skin or eye irritation that's reversible, or we're not going to look at dizziness and drowsiness that are reversible because we don't think there's any potential for that kind of exposure. And then we can ignore aspiration hazards because in order to aspirate, you have to swallow a substance. And in our pretend case, accidental swallowing of the substance isn't anticipated. So we're having a little pretend case where we're doing process development and we have 3 pathways and we have a choice to make between using these precursors, benzene, N-hexane, and nonane. And so this is real simplified, but it's only meant to be an illustration. And so the UN harmonized DHS assignments are where you start. And so benzene's a class 1 carcinogen, class 1 mutagen, and a class 1 toxic organ effects. So N-hexane is category 2 for reproductive toxicity and category 2 for specific target organ toxicity. And nonane doesn't have, it's not UN harmonized. It's not one of the 7,000, almost 7,000 compounds that has UN harmonized DHS assignments. So here we are to the second step of the framework, which is to gather reported toxicity assignments for nonane because nonane doesn't have UN harmonized assignments. And these are the assignments for nonane that are available on the ECHA website. And so 34 entities are reporting that they either import or produce nonane. And these aren't the only hazard categories that they assign, but these are the ones that we've chosen to focus on. So 1 reporter assigned category 3 for acute toxicity for nonane, and 10 of them assigned category 4 for acute toxicity, both of them for inhalation. And in the next slide, I use the category 3 assignment for the toxicity because that's conservative, it's more protective. And there's only 5 toxicity categories in this plot because only 5 toxicity categories are associated with these chemicals. There's way more than that many toxicity categories in DHS. And you can see from this plot that there's trade-offs between inhalation hazard, fertility damage, carcinogenicity, mutagenicity, and organ damage. Already you see the trade-offs. Remember, the 1 is most toxic. So if it's out here, that's the highest category of potency that it can have. And 6 corresponds to no effect. So the spots right at the middle means there's no effect that's been assigned. And these values are the category values. So just from looking at this plot, you might argue that benzene is the worst because it has some pretty terrible effects and they're at the highest level of potency. And then nonane seems like it's the best. It's only a category 3 for the worst thing it does. And the trouble is though that those aren't UN harmonized values. So from here you wouldn't choose nonane, you take additional steps to verify nonane's lower toxicity before assuming that it really is better toxicity wise than N-hexane. And besides which, you only have 34 producers and importers, and that falls way short of reassuring. So there's a lot more work to do on nonane, but from this exercise, you wouldn't rule nonane out. But it is, I think, important for people to accept that if a substance doesn't have UN harmonized assignments, you might have to rule it out just because you don't have conclusive data. So we're back to my takeaway slide. After the development stage, you've committed to the reactants and the carriers, and you've committed to the product and the unit operations you're going to use to make the product. And once all of that is in place, there isn't a lot of opportunity for improving toxicity impacts. And so I want you to walk away from here knowing that any kind of systematic consideration of toxicity is better than nothing. And the framework I presented is only an example. I'm not claiming it's better than any alternative framework. But thanks so much. And I look forward to taking your questions at the end of the program.
Great. Kirsten, thank you so much. And a quick poll result is of the people who voted, no one said that they had, in fact, considered toxicity at all stages of process development. So now we're going to turn it over to Dr. Patrick Harman from BASF. And as Kirsten said, we are going to have Q&A at the end. And there's another poll question. If you, we'd love to hear your responses. So click on the word polls at the bottom of your screen. And the question is, do you think that the current social and political focus on PFAS will result into, and then you have three choices, new alternate solutions, or you think that companies have already resolved the issue, or you think it might not have any effect on product manufacturing. So take a look at the poll and please vote. And now we'll turn it over to Dr. Patrick Harman. Get his slides up in just a moment.
Okay. I think Kirsten might need to stop sharing. Let me see. Oh yeah, there we go. Yeah.
And Dr. Harman, before you get started, it looks like we have some optimists on the Zoom today that most people think that there will be some new alternate solutions coming up for PFAS. We turn it over to you now.
Okay, very good. Thank you for the invitation to talk today. I want to share a few examples of some things that we're doing, particularly in alternatives assessment, and also just some what I see as some challenges to be able to do this properly. So let me, oh, there we go. Okay, so why is it important to do alternatives assessment and to kind of look carefully at safer chemistries? One of these is you want to do informed substitution, and I think Kirsten really touched on this before. Is you want to begin looking at what data are available for the substitutes you're considering. The magician and the rabbit on the side, my colleague in Germany has been using this in presentations going back over 15 years ago. And his point is that very often companies do what he would call alternatives hopping. So they choose an alternative to replace something that has some, let's say, some regulatory challenges. And then 2 years later, it's under the same kind of scrutiny that the substance that it replaced. And so magically, they just start pulling another alternative out of their hat. And so I guess one of my points is that this is not magic. This is really a scientific process that you can use to begin looking at safer alternatives. Secondly, you want to avoid regrettable substitution. And I mentioned this before, you want to avoid reformulation over and over again. This costs money, takes time. There may be regulatory and other costs involved in doing that. Secondly, you want to reduce the risk of future regulatory issues. And I don't think you can 100% protect against this, but I think some of the things that we try to do is to help kind of predict questions that might arise 5 years from now or 10 years from now, and being prepared for that. Also, and part of BSF has a key part of our corporate strategy is based on sustainability. And so and I'll talk a little bit more about this in a couple of minutes, but we have a methodology called sustainable solution steering. And the health and safety of products is one of the ways in which products can contribute to the broader sustainability needs in the marketplace. And I have also enabling recycling. One concern sometimes is that materials, that some legacy materials end up in the recycling streams. And as you move to these newer alternatives, you can reduce the risk associated with that. Okay, so some approaches. One, there's a methodology known as green screen. It has a full set of hazard criteria and I think a fairly transparent methodology. And then one thing I do like about it is that data gaps are not rewarded. So very often I think it's the old term that the absence of data doesn't mean the absence of effects. So there's a weighting of the results based on data. A newer initiative is now what we're involved with is called Chem Forward. And it's was co-created with some brands and retailers. And very often they are the ones that are driving the move toward safer chemistries. And so I kind of like it in that you've gotten some input from the, I guess, from the ultimate market for this. And it's not a new methodology per se. It's a collaborative database of hazard assessments. And it uses GHS and cradle-to-cradle criteria. So really for Chem Forward and Green Screens, there are licensed profilers who do this. And with the GHS and cradle to cradle, there obviously is a lot of overlap, but there are some unique features. Cradle to cradle, can be a little bit more precautionary, which may be important. In the end, products end up, based on the assessments in different hazard bands, A, B, and C, and F. So F are the ones that have more significant, hazard classifications. And really anything in the ABC category are recognized as quote safer chemistries. One of the businesses that I support is our plasticizers business, and so there are 15 alternative plasticizers with assessments, and then two particular for BSF are plasticizers DOTP and DITCH. And then the other thing is important, and I know Kirsten talked about lists. One of the problems with lists is that people will make claims that a product is safer because it's not on a particular list. In other words, it might not be on Proposition 65. So the question always is, well, what do you know about it? And so one of the ones is the EPA Safer Choice Program, the Safer Chemicals Ingredient List. And then there's a private database called Clean Ingredients. So these are substances that meet the EPA criteria that allow people to formulate, with substances on clean ingredients. Our plasticizer, platinum DOTP, is one of the plasticizers that is on this. And I include the last point is that this is taking these approaches. So BSF has a large toxicology department. A lot of colleagues who support our business are toxicologists, so we don't necessarily need this. However, we have to communicate with our customers and also other stakeholders. And then also our customers also need to communicate with their customers. One of our customers told me that, some of these things that we're doing are not requirements, but anything that we do that helps them be able to communicate the benefits of their products to their customers is greatly appreciated. So what does all this look like? And I'll just show a couple of quick examples. So this is what green screen assessment for a DEHP. So this is diethylhexyl phthalate. I don't really want to pick on it, but it's kind of an illustrative example. And all of the letters kind of designate different hazard criteria, or hazard endpoints. So R is reproductive, D is developmental, E is endocrine. And the substance in Europe is classified the substance of very high concern for some of these endpoints. I think more importantly, each of these endpoints, there are data, good, reliable data for each of those. So anything that replaces it should, one, be similarly studied, but also should obviously demonstrate lower toxicity. So one of my, 10 to 12 years ago, a company was promoting a new plasticizer, and we were kind of interested. It has some nice properties. And so my colleague in our technical service group ordered a couple of samples, a couple of different versions. And when he got them in, he calls me up and goes, we took the samples out of the box, and the SDS, had all sorts of warnings that it was severely corrosive to the skin and eyes. And so I began kind of looking at some of the other properties and things like reproductive and developmental effects, low classification, low concern there, but indeed severely corrosive to the skin and eyes. They're also, they had high toxicity, aquatic and chronic toxicity in the aqueous environment. And what this, I think the point I want to make is that, so this is, not an orthophthalate. So if someone wanted to make a claim that it was, safer because it's not an orthophthalate, well, it's kind of hard to make that claim. Also, we ultimately did not pursue anything with this material because you're going from something that, may or may not have some longer term chronic effects to something that has a very real workplace exposure issue in terms of the skin and eye effects. And then also just from a manufacturing standpoint, you have to be concerned about aquatic toxicity. And we did, our toxicology manager did, ask one of his colleagues to do some modeling. And it would have been possible to blend this material with some other products that we have that do not show the skin and eye corrosion. And you would reduce the hazard to just irritating for the blend. So that, I think that also shows some of the potential benefits of screening tools to look at ways you might be able to manage the hazards of the substance. And then this is one of our plasticizers, the DOTP or DEHT. I think the point when you look at all the data, low hazard, quite full data set, the profilers ended up with a conclusion of the data gap for endocrine effects. However, we disagree with that and believe that there is sufficient data to show that there are none. And I have the reference to a publication that we did kind of discussing, and I would say the paper is 95% positive with respect to green screen, but it is a little bit of a critical review of that methodology and of others. Okay, so I mentioned earlier it's sustainable solution steering. This is a BSF methodology. Part of it is assessment of our existing portfolio to differentiate between products that have some significant contribution in terms of sustainability, and then which we call accelerators. A large number of products we call performers, so they may not have any unique contribution, but they don't have any what I would call red flags. They may be global commodities where BSF doesn't really have any particularly unique position. And then we also identify things that are in this, maybe they have some red flags, but it's easily to manage the risk. And then we have what we call challenged, and there's actually a commitment to phase these out over a 5-year period once they're identified. I want to point to the bullet point that's highlighted. And not only is this done to look at existing portfolio, but this process is integrated into our R&D pipeline. So BSF uses, similar to a lot of other companies, a gated R&D process. And so fairly early into the development phase, there are assessments done, one to identify any particular problems, but also to identify benefits. Obviously, if you have something that you want to promote, then this can be very important. Okay, so and this is a slide from a presentation I made last week, and I want to talk a little bit about the challenges. And one of these has to do with data gaps, and particularly when you're in the R&D phase early on, you may not have all of the data points. And ultimately, if you want to go to market, you want to do registrations, you really need reliable, good study data. I mean, the alternatives to that are obviously read across or in vitro data or in silico computational data. And one of the challenges developing all of this are the costs. There was an article in Chemical and Engineering News in 2015. And in one of the interviews, Mark Holt, who works for another company in our market, said, and I won't read all of this, but he said, yeah, no one's going to switch to a non-phthalate plasticizer without a full set of data on PPAR proliferation, testosterone suppression, reproductive toxicity, carcinogenicity, or mutagenicity. The industry is making sure it has all the I's dotted and the T's crossed. And we agree with this. And I asked one of my toxicology friends to put together kind of a rough list of how much all this costs and how long it takes to do it. And you get to $2.5 million pretty quickly. This does not include the environmental testing and also any other special tests that you might want to do for your marketplace. So how do you overcome this if you're trying to bring new products to the market? I mean, this is a sizable hurdle that you may need to reach. And then if I'm going to go back to this closing slide on the challenges, so you have the cost, and I think when you start looking at the weaknesses of things like in vitro data or read across, for example, with the orthophthalates, we know that some of the adverse effects that are driving the regulation are really focused on the alcohol length of the alcohols used to make the respective esters. So if you're smaller than that range or higher than that range, you don't see those adverse effects. So if you only rely on read across, you may get false negatives or false positives. Some of the in vitro tests are really valuable. Some of them are becoming accepted by regulatory agencies, and certainly early on they can give you some indication, but they may or may not be reliable for making final decisions. I think people are aware that I think EPA wants to phase out animal testing by 2030. There's similar directions that are being proposed in Europe. The problem, and I think one of the challenges, is that some of these methods are not fully reliable, particularly when you get into endpoints like reproductive and developmental effects that are really driving a lot of the regulatory attention for some of these products today. So I think this is a challenge for the industry, challenge for the whole world of toxicology to begin, trying to develop reliable methods. So certainly that can be used early on in the development phase to help make sure that we're creating the right types of materials in the future. So quick summary, data are important. The absence of data doesn't mean the product is safe. And I think we need to be focusing on making sure that we have data on our substances, but also developing new ways where we can get this information faster and earlier on in the process at some reasonable cost. Okay. So.
Thank you. We really appreciate all your insights, and we look forward to the Q&A. We already have some questions coming in, and people, if you have questions, please type them in the chat. We'll answer as many as we can. And now we're going to hear briefly from Viridis Chem CEO Neelam Vaidya, and then we'll do the Q&A. So over to you.
Thank you, Laurie. So today we heard from both the panelists about the importance of the toxicity information and the reliability of the data, and as well as the limited information, experimental data available. And hopefully Viridis Chem can answer some of these questions, and I would like to discuss how. So can everybody see my screen? Hopefully all of you can see the screen. So here today I would like to discuss very quickly the software platform Viridis Chem has, and the product chemical analyzer that offers detailed toxicity analysis of every chemical, whether it's known, unknown, or even the new molecules. So first, the platform. Viridis Chem has built one of the largest toxicity database with 93 million chemicals, 3.5 billion properties, and it has incorporated information from more than 200 different sources, including government and research organization databases, 136 different regulatory lists, material data sheets from reputed manufacturers, as well as the research papers and data obtained from the chemical manufacturers. We have been curating this data and updating this data using our own internal AI-based tools. Whenever there are data gaps, Viridischem utilizes available industry standard prediction models to generate in silico data. It has the ability to run these models in real time on demand so that we can provide the toxicity analysis of the novel molecules. Using the chemicals, functional descriptors, and classifications, it pre-selects the models. The models include fragment-based, descriptor-based models, statistical models, expert systems, and the biological activity-based models. And we have also built our own read-across models for some of the predictions. Using the OECD guidelines and available properties, we have developed our own proprietary algorithms to provide ecological, health, and safety scores that are valued from 0 to 4 so that we can easily show you the comparison between chemicals. Simply bigger the area and then the graph, more toxic the chemical is. So here are all the different properties that are available through our product. We have developed our, when our experimental data is not available, we select the in silico data that we have developed using these models. And here are the different scores that we provide based on the physical and toxicological properties. The scores are based on ANSI 355 standard and include more than 56 different endpoints. So the information that we provide is pretty comprehensive. So let's talk a little bit about our product chemical analyzer. It's used by the drug discovery groups to understand the toxicity of new or unknown molecules and explore their analogs and metabolites. By product development teams to understand the toxicity footprint of multi-state processes while selecting the best reaction route, and to find less toxic alternatives that can replace the toxic chemical during product optimization. It's also used by the EHS and product stewardship groups to understand the global regulatory implications and occupational hazards, and by the lab safety managers to isolate fire hazards, acute health hazards, and other safety hazards. One of the key features the product has is its advanced search. And here is how it works. If you know a toxic reagent that must be avoided during the product development, identify its role in the reaction and the critical properties that are needed for the successful reaction. Using these properties along with what you would like to have or to avoid during the in the new chemical, use that as a research criteria. And these can be physical properties, value range, or specific functional groups that you want or do not want, or any health issues that must be avoided. Then select 2 or 3 best results and compare them to select the best one. So now I would like to discuss specific examples. The first one would be the select the least toxic options. How do you do that? So until recently, textile industry was using formaldehyde, a strong oxidizing agent, to create wrinkles in the clothes, as well as to create develop wrinkle-free clothes. But since formaldehyde is very toxic and volatile, researchers wanted to find less toxic alternatives. The group GDHC identified 3 alternatives that are oxidizing agents: zinc formaldehyde sulfoxylate, thiourea dioxide, and sodium dithionite. So we compared these chemicals, and as you can see, zinc formaldehyde sulfoxylate seems to be a better choice among the 3 chemicals. The second example would show how to find the toxic alternatives that satisfy your reaction needs. The goal here is to replace the iodomethane, a volatile, costly reagent that generates toxic byproduct iodine, with a better alternative chemical. So we know that the reaction needs alkyl carbonate, alkyl carbon, and alanine side chain as a functional groups, and we'll also include carbonyl group so that the byproduct generated will be less toxic. So our advanced search criteria would be boiling point greater than 60 degrees and vapor pressure greater than 40 millimeter of mercury to find less volatile chemicals. We want to select functional groups, alkyl carbon and alanine side chain, because they are needed for the reaction, and carbonyl to ensure the less toxic byproduct is generated. We also want to avoid the health issues, eye irritant and skin irritant. So here are the results. Out of these results, we selected 1-methyl, 1-methoxycarbonyl ethyl and dimethyl carbonate because of their low health scores. Now comparing the two selected alternatives, we selected the dimethyl carbonate because it's less volatile, less toxic, with lower eye and skin irritation scores. And it also happens to be less expensive and generates environmentally benign carbon dioxide. So I hope you like what you saw. Please contact us if you'd like to have discuss your company needs and for a quick demo from our team. Thank you.
Thank you so much. So we're now going to switch over to Q&A, and we have a couple questions already. First, Dr. Harman, we have a question for you. You mentioned this term cradle to cradle, and some of us have heard of cradle to grave, but we wonder, can you explain what you mean by cradle to cradle criteria?
Yeah. So you might look up Michael Braungart and Bill McDonough. And so they kind of initiated this process, and it's spun off into its own organization. But they do assessments on materials mostly for the building and construction area. And the idea is that, we in the past have designed materials cradle to grave, but with all the discussion about circularity, things can be designed of how they fit back into this whole circular economy. And they were talking about this, 15 years ago, when they were kind of working on this. When I mentioned it in the talk, Kim Forward uses so the Cradle to Cradle organization, they have a material health assessment criteria, and so that particular methodology is used for the assessment of chemicals for this. But the term cradle to cradle, I think, goes back to Michael Braungart, who's a German chemist who's still active out in the marketplace.
Okay, well, thank you. And a quick question for you, Kirsten. One attendee today wondered, could you explain further the difference between data gap and having no data, and what should one do?
Yeah, I think Patrick covered that much better than I did. So a data gap is when you have no data. And in the past, I think we've seen a lot of examples where, what did Patrick call it, where you're hopping substances. People treated a data gap as if that was protection against toxic effects and other effects. And so we know better. We've known better for a long time. If you have a data gap, it doesn't mean that a compound is benign.
Right, and you said earlier that assume the worst.
Assume the worst.
Yeah, wow, that's memorable. And here's a question for you both that's coming from an attendee. The question has to do with trustworthy sources of data. And this person commented that they feel that lists like the classification and labeling inventory from the European chemicals agencies are not reliable, and that material safety data sheets often are not reliable. So what should be the most trustworthy go-to data? Is it the GHS? And we'd love to hear from each of you.
Yeah, I'll talk first and let Patrick go. I think that the UN harmonized values from GHS have been curated, and they've been curated by people who know what they're doing, and it's all very transparent. The CNL, it's a law that people understand the toxicity of the chemicals that they're importing and producing, and that they convey that information on these globally harmonized safety data sheets. But that doesn't mean that they're doing a good job of it. And definitely, you have to use that information with caution. I think it's more like you can rule a compound out than you can decide that it's in based on that information. So Patrick is way deeper into this on the proprietary database side than I am, and he can talk more.
Yeah. Yeah, that's a good point, and I think if I read the question correct, or I read the question or point correctly, is that some SDSs and some classification and labeling decisions are good, they're accurate, but then some are not, and so how do you move past that? So one of the things I know with REACH, you have the registration dossiers, which are robust summaries for the different endpoints. I mean, those can be helpful if you want to kind of read in greater detail. And then you can also see what data are missing. Very often there are substances that the decision is made it's not necessary to include a particular study. But if you're trying to do a full assessment, maybe it would be nice to have that data. And I think this is why you're seeing a number of people out available, consultants who are providing their services to help companies kind of do those assessments. And in some cases, if you have a supplier that you're working with, they might be able to provide the information confidentially to those consultants under an NDA to be able to help make those types of reviews.
So I would like to actually add to that. In our experience, while we were collecting all this different data, we realized that even the experimental data, a lot of times it's not aligned with each other. It's different because so much depends on the environment in which that experimental data was created. And we also realized that certain types of data would be more reliable from one sources for the others. And so it really depends on the context of the data. We could not identify one single source that was better. So we ended up really, defining the hierarchy with the most trusted data for this type of data, chemicals, and relied on that hierarchy.
Yeah, people should be aware that with toxicity data, the uncertainties are huge. If you do animal testing, there's variety. Even when they use very similar strains of animals, there's variety from animal to animal. And then even within the same species, if there's a different strain of mice, you might have totally different responses. So when you jump from mice to humans, or from rats to humans, you're merely making a leap. And I don't know if anybody has noticed in the news in California, there's some very recent finding where women who were pregnant and lived within 2 miles of certain pesticides being applied give birth to children who are much more likely to have childhood cancers. So how are you going to identify something like that in a toxicity study? The most tragic cases are the ones where we have human data. And I think, it's a tough problem to solve. It's a really tough problem to solve.
Yeah, wow. Well, thanks all of you for addressing that one. Oh, did you have something more to say?
Yeah, just a quick question. I think also this is one of the reasons that you may not always have the answer to every question today, but I think the responsible way is to try to answer those questions as best as possible. And I know that we've been involved in work trying to understand things like metabolism of substances and how they compare in humans versus animals to help kind of bridge that gap and understand of is the animal model is it pretty good or is it weak, and So I think those are, I think open areas where a lot of research is still needed for a lot of substances.
Mhm.
Wow. Okay, well, here's another question for you all. So we all know that companies are paying a lot more attention to the toxicity of chemicals than they were 10 years ago. And beyond what you've said today, or if you want to emphasize anything in particular, what advice do you have for scientists about simple measures they can take to minimize toxicity footprint during either product development or manufacturing?
Well, my argument would be to do something, something systematic and reasonable from the very beginning of the process development, and really consider what product you're developing and all the steps along the way. And Patrick has gone through some really specific examples of that too.
Yeah.
Pick a scheme and apply it. It's much better than nothing.
Yeah, and I don't know that everybody is doing this to the extent that they should, but I think it's becoming increasingly important to have. So if you're a chemist or a chemical engineer, you're making a new product, developing a new process, to also early on engage toxicologists and other people and other particular functional groups to help answer those questions early on. You don't wait all the way to the end. It's like, oh well, now we have a problem, and we're trying to get this product approved, and we can't because we didn't look at this or that. So I think part of it is having some engagement with people who kind of understand those potential concerns fairly early on in the process.
Yeah, and there's the time pressures are intense. Patrick was talking about an 18-month delay to get all the toxicity information, but a lot of times if your competition is going ahead without doing that and they beat you to the punch, you've lost your opportunity. So it's really tricky. You have to decide what your priorities are and what chances you're willing to take. And the consequences can be so bad.
Absolutely. Wow. Well, thanks, both of you. Well, it's almost the top of the next hour, so I'm going to turn it over to Neelam for a few words to wrap us all up for today. And thanks everyone for joining. Over to you, Neelam.
Thank you. Thank you, Laurie. Thank you everyone for joining the webinar. I hope you will be joining the future webinar. As I mentioned before, we are hoping to have 5 more webinars on this particular green chemistry. What does it mean for the different industry sectors? And we will be announcing the next webinar soon, hopefully this year rather than early next year, but definitely the December or January.
Great.
So looking forward to seeing you again.
Yes, absolutely. And everyone, we have one more final poll question for you. We'd love for everyone to vote. We'd like to know, we seek some information for you to finalize the planning on our next webinar. So if you'd please take the poll question, we would really appreciate that. And thanks again to our speakers, Kirsten